Good afternoon and welcome to this event at De Montfort University, part of our distinguished lecture series focusing on Nobel Prize winners. This lecture series was initiated by Professor Parvez Harris in 2017 on behalf of the university and was based on the firm belief that Nobel Prize winners can be engaged to promote greater interest in science and education. And this cannot be more so for an institution like ours, where widening participation is key to our ethos of supporting social and economic mobility. Making these prized individuals more accessible to our students and researchers and the general public will certainly create a greater appreciation of science and also inspire the next generation of leading scientists and hopefully one day a Nobel Prize winner. The first Nobel Prize speaker in the series was Sir Paul Nurse, a Fellow of the Royal Society and 2001 Nobel Prize winner in medicine. The second was a lecture delivered by Sir Gregory Winters, a Fellow of the Royal Society and the 2018 Nobel Prize winner for chemistry. Today we have the third speaker, Sir Richard Henderson, Fellow of the Royal Society and 2017 Nobel Prize for Chemistry. We're really honored and appreciative of the time of Richard Henderson in giving to us this afternoon, not just his time, but also his thoughts. And I'm really looking forward to hearing his lecture. I will now invite Professor Parvez Harris to introduce Richard Henderson. And thank you once again for joining us.
Thank you, Professor Ma, and I would like to welcome everyone from around the world who are joining this distinguished lecture series. A lecture to be given by Richard Henderson. Actually, he told me not to call him sir. He told me not to call him professor. He just asked me to introduce him as Richard Henderson. So he's a very humble and, of course, a very hardworking scientist. First of all, I would like to start with giving some context to his presentation. Because myself, like him, has been working on proteins. And proteins are the workhorse in living organisms. They are the molecules that do things in our body. So for example, when you eat food, the food are digested by the enzymes, which are proteins. And when infectious agents enters our body, like for example, the coronavirus or any other bacteria or any other pathogen, it is the proteins that actually go after them and try and neutralize them. So we heard about proteins a lot, especially during this pandemic, because we know that the coronavirus itself is able to enter the human cells through its spike protein. We've seen these images in the television where you see the spike protein protruding out of the coronavirus. And it is this protein, which is a membrane protein, a protein that is embedded in the lipid bilayer or an oily lipid environment, fatty environment of the cell, outer layer. And that protein actually enables the virus to enter the cell. So the proteins are the critical players in our body. They are involved in diverse biochemical processes. So when Donald Trump got those experimental treatment, that included actually antibodies. And antibodies, of course, are proteins. So proteins are really very important for many, many different processes in living organisms, not just only humans. And what I really need to say is that proteins are of many different types. There are thousands of proteins in our body, but there are two main classes of proteins. One is membrane proteins, and the other is soluble proteins, water-soluble proteins. So the water-soluble proteins are the ones that are readily soluble in water, and they can be easily purified, and they could be easily analyzed. When I say easily, relatively easily. But the membrane proteins, because they reside in a very complex environment of the membrane, very oily, fatty environment, they're very difficult to isolate, they're very difficult to characterize, and it's extremely difficult to get a membrane protein structure. And today, we are really, really honored to have the first person in the world who has actually obtained the three-dimensional structure of a membrane protein. And that's very important, because when you know the architecture of anything, you know how to control that particular object or system. So proteins, when you know their three-dimensional structure, their architecture, then you are able to control their activities and functions. So if you know the structure of the coronavirus spike protein, then you can design vaccines and drugs to prevent it from infecting human beings. So knowing the structure of proteins is pivotal for understanding their function and also for designing drugs and vaccines. And it is for this reason, I think, that we have to be very grateful to Professor Richard Henderson. I should just call him Richard Henderson, just call him Richard. And he actually is a man who was the first in the world to crystallize a membrane protein. And when I talk about crystallizing, getting the structure of a membrane protein, I have to say there are proteins and there are proteins. So he looked at membrane proteins, the most difficult class of proteins, which most people fear to actually work on. Many scientific lives have ended without any success in getting the structure of membrane proteins. And many have sort of not even dared to look at membrane proteins because of the complexity that it involves. But Richard Henderson wasn't afraid of that. So, you know, I try to provide an analogy. Water-soluble proteins are like mountains that are easy to climb, relatively easy to climb, like Ben Nevis or Mount Snowdon in Wales or Ben Nevis in Scotland. Whereas membrane proteins are like the equivalent of the most difficult mountains to climb in the world, like, for example, Everest or K2. K2 is also known as the Savage Mountain, and not many people dare to climb it because most people die trying to climb it. So in the sense of protein structure, I would say that Richard Henderson is really a brave and courageous man to have embarked on a scientific life that involved tackling the most difficult proteins that one can imagine, membrane proteins. And he has been successful in being the first person to elucidate the complete three-dimensional structure of a membrane protein in its native environment. And I like to stress native environment because when you remove a protein from its natural environment and membrane environment, then you're not really looking at it in its real biological environment. And what Richard Henderson has done by developing the technique he has developed is that he has been able to look at the three-dimensional shape and structures of membrane proteins in their physiologically relevant environment, looking at them in the membrane, in the cell membrane, and that is unique. And it is not a surprise, therefore, that he was awarded the 2017 Nobel Prize in Chemistry for his work developing a technique called cryo-electron microscopy, which enables one to look at proteins in difficult environments, including in membranes. So it is no wonder that he was awarded the 2017 Nobel Prize, and it's a well-deserved winning of the prize, which is shared with Jacques Dubochet and Joachim Frank. And really, again, I am really honored, and the university is honored. We are all honored. All the audience that are listening to this presentation are honored, because really it's difficult to get such a great speaker to come and interact with the public. And what work that Richard Henderson has done is such an important topic in the current situation. We are all faced with the pandemic, COVID-19 pandemic, and his research work on the structural characterization of membrane protein using cryo-electron microscopy is paving the way for us to understand the structure of the coronavirus protein, the COVID-19 protein, and thereby developing better drugs and better vaccines. Here I have a slide where I'm trying to compare his work with climbing mountains. When Richard was a PhD student back in Cambridge, he actually elucidated the structure of a water-soluble protein. And I give the analogy of climbing Ben Nevis, which, by the way, Richard has actually climbed. He told me that he has climbed Ben Nevis. So solving the structure of a water-soluble protein, chymotrypsin, was a relatively easier task because it's a soluble protein. You can crystallize it easily and you can get its structure. However, getting the structure of membrane proteins is much more complicated. And if you look in this slide, you can see I have presented on the top panel as water-soluble protein, chymotrypsin, and its three-dimensional structure solved using X-ray diffraction, equivalent to climbing Ben Nevis, which is, by the way, the highest mountain in the British Isles. And then beneath, I show you the membrane proteins. There are several different membrane proteins here, but I like you to focus on bacteriorhodopsin, a protein I've also worked on. And this is a protein that is a proton transfer. It's a membrane protein from a bacterium. And Richard was the first person to actually solve the three-dimensional structure of this amazing protein. And this, as I already told you, is like climbing the deadliest mountain on the planet. And this is K2, which is known as the Savage Mountain because of the number of people who have died trying to climb it. So I really, really think that his achievements are noteworthy. And indeed, the point I like to stress is that there is a difference between reaching the summit of a mountain and solving the 3D structures of membrane proteins. If you look here in this slide, what I'm saying is that reaching the summit of K2 gives one self-satisfaction and human victory over mountains. It is a great achievement, and it's something all human beings enjoy, and we see that we can conquer toughest mountains in our planet. But what Richard Henderson has done is that he solved the 3D structures of membrane proteins. And the impact of this is far wider. And it goes beyond the personal self-satisfaction. It goes into helping humanity at large, paving the way for producing life-saving drugs and vaccines. So without taking more time, we like to listen to Richard Henderson, an amazing scientist, an amazing mind, and we are extremely lucky to have him here. I very much hope that you will enjoy listening to him and asking questions at the end of the presentation. So without further ado, I'll pass it on to Richard for his talk.
Hello there. I don't think some of us couldn't see your two slides, Parvez. So maybe we can see them later. Let me know if you want to change. Anyway, it's a great pleasure to join all of those who are watching. It had been planned by Parvez that I should visit De Montfort during the year 2020, but we've been sort of snookered by the coronavirus. So it is still 2020, and so it's delightful to come and join you. And thanks, Parvez, for all those kind words of introduction. So now I'm going to share my screen so that you can see my slides, and that might take a moment or two to get it. Allow. Let me see if that comes through. I can't see it yet. Yes, now you can see it. So now I'll switch to the full screen view. So hopefully now, please interrupt me if you know if something goes wrong. Holly or Parvez, who are able to see this, if it's not working properly, let me know and I'll go over it again. Okay, so the idea was to give a talk that was a general overview, so not only suitable for people who are structural biologists, but to try to give a general introduction first. So the talk I've got consists of a sort of mini history of structural biology, starting with X-ray diffraction, which was started back in the early 1910, 1912 by von Laue and Bragg, and then talking a bit more about electron microscopy, NMR, model building, and so on as an introduction. Then most of the slides and most of the talk that I'll present is about cryo-EM, because it's been around for 30 or 40 years. It's become more and more powerful, and now it's very powerful with a great future. So it's important to tell you about that. And then Parvez especially asked, so I've been spending a little while catching up with all of the great work many people have been doing during this last year on the coronavirus, on SARS-CoV-2. So I'll end up with a few slides about the different structures in SARS-CoV-2 and how they impact on health and the viral infection and so on. Okay, so the first slide then, see if I can get this. The first slide is an overview of structural biology from before any biological atomic structures were known, really. And in the beginning, of course, X-ray diffraction, electron microscopy, nuclear magnetic resonance, but they weren't very powerful methods. So people began by doing model building. And I'll tell you a little bit about Linus Pauling's original work, and Rosalind Franklin and Watson Crick and so on. And then a bit about X-ray crystallography, one slide only about NMR, and then most of the talk will be about electron microscopy. So here is a slide of Linus Pauling, who won the Nobel Prize in chemistry in 1954, and later on in peace for his anti-nuclear weapons efforts. And he's pointing at the model he made of the alpha helix, which was built on paper and with models based on his work using X-ray crystallography, studying the structures of amino acids that proteins are made up of, and particularly dipeptides. So he was able to show that the proteins had certain structures, the peptide bond, that were planar, and that allowed him to come up with the alpha helix and the beta sheet as being what he proposed as being the underlying structural motifs in all of proteins, in all of biology. And he was right about that, and that was his original contribution. The second slide is the famous work on finding out the double helical structure of DNA, which underlies all hereditary and viruses and so on. This is Rosalind Franklin, whose work at King's College London with her student Raymond Gosling ended up with them taking this beautiful picture. This is called Photo 51 of the hydrated B form of DNA, and this cross shape with the dense structure up here along the axis allowed Francis Crick and Jim Watson to come up, piecing that information together with lots of other information to come up with a double helical model of the structure of the DNA double helix. And so in the beginning of structural biology, the structures for proteins and for nucleic acids, DNA and RNA, were all worked out by cobbling together information from a wide range of information and then doing model building. And that was the beginnings of structural biology before the experimental methods became very, very powerful. So then, switching to the second stream, these are beautiful crystals of sperm whale myoglobin, which was John Kendrew's work that he began when he was a PhD student with Max Brutz here at the MRC Molecular Biology Labs, the previous unit before the lab was called by its current name. And they actually isolated myoglobin from numerous species, horses, cows, cats, dogs, as well as seals and whales and sturgeons and so on. And these were the ones that were the most suitable for the early methods of X-ray crystallography, which needed crystals to have a certain size and shape to be amenable to the method. And in the 1950s, this was their very first, and it's the very first model in 3 dimensions of any protein. It's myoglobin, a model made in 1957, made out of plasticine, and it's been colored white as these sausage shapes, and then this disc was actually painted red. That's the heme group that gives the myoglobin its color. It's the iron in the heme group is the site at which oxygen is taken up by myoglobin, and of course hemoglobin in the blood for transporting oxygen. And these sausages are all about 10 angstroms apart, that's 1 nanometer apart, and they have the dimensions, the same dimensions that Pauling's alpha helix had. And it turned out they're all alpha helices, and one of the major motifs, this was the first time it was seen, is this alpha helical structure. And in membrane proteins too, most of the structures in membranes are made out of alpha helices that cross the membrane from one side to the other. So in John Kendrew's group in the 1950s, they went on to collect more and more data and get to higher resolution. And this is a structure, an atomic model of the myoglobin, the first protein structure, the only one that was known. They found out the structure in 1959, 1960. There wasn't another structure until 1964, which was hen egg white lysozyme. And this is a painting made by Irving Geis, who was an artist that John Kendrew persuaded to make this watercolor painting, which was in the center page, double page spread in the middle of Scientific American in 1962. And, you know, the artist has changed the position of the atoms ever so slightly so that you can get so that the front ones don't occlude the back ones. But this and another painting he made a few years later of lysozyme are essentially the beginnings of protein structure in 3D done by experimental X-ray diffraction, but represented by an artist painting each bond, each atom now, of course, we have beautiful molecular graphics that can do it much more quickly. This is, on the left, John Kendrew, who was the head of the group that worked out the structure of myoglobin. On the right is Max Perutz, who was working at the same time on the structure of hemoglobin. And Kendrew was Perutz's PhD student. Perutz himself was supervised when he did his PhD about 20 years earlier by Lawrence Bragg. And Lawrence Bragg was an artist also, and he made these two charcoal sketches of his two essentially structural biology disciples, following on from Bragg's work, developing X-ray diffraction as a method of analyzing the structure of crystals. And for that, Bragg himself, with his father, shared the 1915 Nobel Prize in Physics, whereas Perutz and Kendrew shared the 1962 Nobel Prize in Chemistry for their work on hemoglobin and myoglobin. So if we go on, this is Max, a bit older, with his model at the same lowish resolution as myoglobin, as it was in 1960. So that's a little bit of a snapshot of the origins of structural biology as done using X-ray crystallography. Another method came into prominence in the 1980s, and that was the indirect determination of protein structures using a spectroscopic method, nuclear magnetic resonance spectroscopy, that Richard Ernst developed with Fourier transform methods in the 70s. But Kurt Wuttrich, who was in Zurich for many years, translated those early methods and made them powerful enough to determine the structures of proteins. And this is a 2D nuclear Overhauser spectrum of protein CD59 using the methods that Wuttrich developed. This is the structure of CD59. So NMR is a good, if you can't crystallize your protein and it's of the right size, NMR can give you the structure. But because other methods have become more powerful now, NMR has now peaked out and is declining. But nevertheless, at the time, from about the year 1982 to probably 2005, it was very popular for determining the structure of things like zinc fingers binding to DNA that other methods couldn't reach. So then we come to electron microscopy, and one of the earliest papers that showed that it was possible to determine 3D structures from electron micrographs came from this work of David Desrosiers, who was a postdoctoral fellow from the USA working with Aaron Klug in the 1960s. This is a paper in 1968. Let me change my pointer to a laser pointer. And so here on the left is a micrograph with a virus, T4 bacteriophage, embedded in negative stain uranyl acetate. And by doing computer image processing of these images, they were able to get a picture in 3 dimensions of the arrangement of protein in that tail. Very low resolution. They didn't really know what the proteins were, but nevertheless, it was the beginnings of 3-dimensional analysis of 2D images that were taken by electron microscopy. And that work was one of the contributors to Aaron Klug's Nobel Prize in 1982. So now we come with that background in structural biology. How did we get going in all these methods? The next slide is one that Tim Baker and I made in the year 2000 and was published in the International Tables for Crystallography. And our idea was to give people an overview of what is electron cryomicroscopy. And I'll say a lot more about this, but the general idea is you take your molecule of interest, and in the left-hand top panel is a picture of the E. coli 70S. That's the full ribosome, site of protein synthesis, done in the group of Joachim Frank. And in the year 2000, when we wrote this review, the resolution of the analysis of the structure of the ribosome was about 11 angstroms. And here is their 3D model on a scale about 10 times bigger magnification than the micrograph. So here you have all the different views of the ribosome. In the computer, you can interpret all those views in terms of the large subunit, the small subunit, The green is a tRNA, and then there's a long RNA helix in the middle of it. And that was a single particle in a thin film of ice that I'll tell you a bit more about Dubochet's plunge-freeze method in a moment. Single particle without any symmetry. The second panel shows hepatitis B virus cores. This is the core of the hepatitis B virus. And this was work Bettina Botcher did when she was a postdoc here at the MRC, Laboratory of Molecular Biology in Cambridge. And this was the first time any structure of a single particle without it being in a crystal was determined in biology at a resolution below 10 angstroms. So that's sub-nanometer resolution. These virus cores have these protrusions, and you can see that there are little ridges along each of the protrusions, and each of those protrusions is a bundle of 4 of Linus Pauling's alpha helices, just resolved at 7 or 8 angstrom resolution, because the helices are about 10 angstroms apart. And essentially, this method, the single particle with no symmetry and single particles with symmetry, without any crystals, is the main method that's being used nowadays in single particle cryo-EM for all the coronavirus and so on. I'll say a lot more about this. But the third panel also shows that you can take images of protein aggregates that are arranged into filaments. And this is the thin filament in muscle made up of actin molecules, decorated by subunits from the thick filament that has the myosin molecules. And these 2 filaments pull against one another and cause the muscle contraction that is in all your muscles in your heart and so on. And Ron Milligan's group, around about 1995, analyzed by cryo-EM the structure of many of these filaments. And they did by plus and minus methods. They could get in blue the central core of actin, in green the tropomyosin molecule that helps to hold it together, and then in pink, orange, and yellow are the different domains of the myoglobin heads that bind to and pull the myosin filaments along the actin and cause muscle contraction. And so even at 30 angstrom resolution, very low resolution, can't see atoms or anything like that, it still told you a lot about muscle contraction. And then the fourth panel is how I made the transition from being an X-ray crystallographer to being an electron crystallographer. Is an image, you can't see any detail really, of a 2-dimensional crystal. And this is the light harvesting complex from green plants from Werner Kuhlbrandt's work, published in 1994. And because there's so many copies of each of the biological structures in the 2D crystal, it's much easier to get to high resolution. So this is a 3.4 angstrom structure of the light harvesting complex with in each protein molecule, there are a trimer, 3 protein molecules, but in each of them, there are 13 or 14 chlorophylls and 3 transmembrane helices of the same alpha helix that Pauling had predicted. And our work a year or 2 earlier on bacteria rhodopsin, was the one that Parvez referred to, had 7 transmembrane helices, and we had a structure for that in 1990, but I'm not going to say any more about that. So I'm now going to say, I'm going to jump from this overview, this introduction to how cryo-EM works, to say a little bit about how these specimens are made before they're put into the electron microscope. And then I should add one other thing before I move from this slide, and that is that if we were remaking this slide now, we would add a 5th panel to include not just regular structures that are repeating, like 2D crystals, helices, or single particles all identical, but you can also do electron cryotomography, where there is no real repeating structure and you're looking at a complete mixture of things in the cells. That is tomography of the same type that you do if you are, let's say you go to a hospital, you have a cancer tumor somewhere in your body, in your brain, or in the gut or so on. You can have an X-ray 3-dimensional tomographic scan done so that the doctors can see where your tumor is. And you can do the same in cryo-EM, except you're using electrons rather than X-rays. And I will come back to that a little bit at the end when we talk about the coronavirus. Okay, so this method of making these thin films, including the molecules, was developed by Jacques Dubochet, who was one of the people who shared this 2017 Nobel Prize in Chemistry. What he developed with his colleagues Alistair McDowell and Mark Adrian was a method for making a thin film of fluid, of buffer, of water, into which you've got your molecules of interest. So enzymes, ribosomes, viruses, muscle proteins, anything you like. And what you see here is a pair of fine forceps, and on the end of it is a 3 millimeter thin electron microscope grid with holes in it. And the method consists of putting a little drop of your solution on that, blotting it with filter paper, and then plunging it at a speed of about a meter per second down into this little chamber. And in it, you have liquid ethane at a temperature just above its freezing point, so at about minus 185 degrees centigrade. And it's cooled by liquid nitrogen around it. And this procedure, putting a drop of liquid on a grid, blotting it with filter paper, and plunging it into liquid ethane at the temperature of near liquid nitrogen, was the method that Dubochet developed in the early 1980s. And we still use it. It's been the basis of the cryo-EM revolution. This one in the middle was a gravity-driven plunge freezing apparatus. But at one stage, they thought they might like to make them go a bit faster. So here on the left, you see another pair of forceps, but instead of gravity, they've got elastic bands helping to make it go faster. And these cost very little money to make. But now, of course, you can buy a computer-controlled plunge freezing apparatus for about £50,000. And many people have these and like them rather than the manual versions. So one of the first specimens that Dubochet and his group recorded was of the adenovirus, which gives you colds and things like that. And there's a slightly interesting story. So Dubochet was employed by John Kendrew, the first director at the MBL in Heidelberg in 1978. And when the second director came in 1983 or so, 1982, 1983, Leonard Philipson, he said he would like some of the people under the previous regime to leave. So Dubochet was given his notice to leave. But then Leonard Philipson had worked on adenovirus for his whole life. Dubochet then took this picture and was immediately reinstated and stayed on at the EMBL till 1987. So he was 9 years there and then went to Lausanne. But this picture was taken in 1984 and was on the front cover of Nature. And everybody who saw that realized that this method was going to have a great future. It just needed to have lots and lots of technical developments. In fact, this was how it stood, the picture alone, and you could look at it by eye until 1991. So 7 years passed until Phoebe Stewart, who is a postdoc at EMBL, in a different group led by Steve Fuller, took images like this and worked out how to convert them using the computer programs, the same type of work that David DeRosier and Aaron Kluger had done, but applied to arcosidroviruses, and got this 3D map of the structure of the adenovirus. So they've got little spikes coming out of them, but they're very blurry, and the resolution is 35 angstroms. So you couldn't really see the subunits, certainly could not see the atoms. And the reason for that is that the images look nice, but as you look at the finer features, they get fainter and fainter, which is why it's a bit blurry. And so that's how it stood until about 20 years later, until new microscopes came in. And Hong Cho, who's at UCLA, took this picture of the same adenovirus in the year 2010 with using a newer technology, higher voltage microscopes, brighter sources, and so on, more stable. And you can see now, instead of it being a bit blurry, you can see a lot of fine detail in each of the pictures of these virus particles. And by processing these in the computer using similar programs to the ones that had been used earlier, they got this structure here on the bottom left, in which the detail you see is very much finer. And then if you blow up one subunit, here it is, you can see that you can see the individual amino acid strands, and the resolution is now 3.6 angstrom resolution instead of 35 angstrom resolution. And these strands of beta structure are about 4.8 angstroms apart. You can see them, you can see the side chains. And so we've gone from blobby structures to near atomic resolution structures by improving the hardware involved in electron microscopy. Another structure done at that same time is the one I mentioned already. This is Bettina Botcher's work. But her earlier work, without the benefit of the modern technology, gave her, from these pictures top left here, roughly a 30 angstrom structure in which there were two different diameters, big ones and little ones, T equals 3, T equals 4 structures. And the one that I showed earlier in that collage illustrating how cryo-EM work, that's this 7.4 angstrom structure with these protrusions, which consist of a bundle of 4 alpha helices. And that's how it stood, again, for about 25 years, until Hong Cho, again at UCLA, took some better images of these and got it to a resolution of 3.5 angstroms, just like the adenovirus. And now you can see the alpha helices sticking out from the surface with individual helices and individual detail, the turns and the side chains and so on. So those were two early examples. But the thing that made the big difference was that new detectors came in about 7 or 8 years ago, 2012, 2013. And this is a technical analysis of the power, the signal to noise, if you like, using a variety of detectors. In black is film, which is what we've been using from 1970 right up to 2012. Detective quantum efficiency means you detect with 0.3 here. It means you detect about a third of the electrons, and the other ones are not detected. And there were no electronic detectors better than that until these new, they're called CMOS, complementary metal oxide semiconductors. They came in, but they took a long time to develop. And the 3 companies that made them, Gatan, FEI, now Thermo Fisher, and Direct Electron in California, brought out these detectors about the same time. And this made a quantum step forward in the power of the methodology. And so I'm just going to show you the sort of images that you got with these new detectors compared to film. So here is some work that Vinod Kumar, who was a postdoc with us in Cambridge for until about 2 or 3 years ago, and Vinod is now the leader of the cryo-EM facilities in India, in Bangalore. But he took all of these pictures that I'm just going to show you. So this is a 1.5 million molecular weight icosahedral particle that's an enzyme, pyruvate dehydrogenase. And here's the ice. So this is the amorphous ice. Here's amorphous carbon, and this is a hole in the carbon blotted by the Dubochet-Plumb-Fries method. And because these are icosahedral particles, you can see 2-fold views here, 3-fold views here, and 5-fold views here. And it's got 5, 3, 2, 7. But you get all the other orientations, and so you can use this and get a much better structure. And you can also take pictures of smaller objects. This is a mitochondrial membrane protein called complex 1. It has no symmetry, like the ribosome has no symmetry, but this one has 45 different polypeptide chains in it. And until these new methods came along, the arrangement of how they're arranged wasn't known. And I'll show a little bit more about this in the future. This is beta-galactosidase, which is a tetramer of 4, something like 110, 120 kilodalton subunits. And by looking at these images, you can see, for example, here, there are 4 little dots that are the 4 subunits of the beta-galactosidase. And beta-galactosidase was the first enzyme whose operon was discovered back in the 60s by Jacob and Monod, who had a Nobel Prize for that work, identifying operons. But in this image, you can see all the different views of the particle, and that's allowed people to get really good high-resolution structures of this nowadays. And then a little bit smaller, this is human erythrocyte catalase, an enzyme that keeps the hemoglobin from being oxidized. And again, the blotting has squeezed out all the molecules from the middle of the little droplet. So this is just a thin film of amorphous ice that's probably less than 100 angstroms thick. And then the molecules have been squeezed out. They tend to form ribbons here and all in one orientation. And then you get other orientations. And then eventually, when the ice is thicker at the edge of the hole, they're beginning to overlap. So that's some pretty pictures. So I'm going to show you 6 examples of the power of the cryo-EM methodology for finding out the structures in biology. And I've picked them to be illustrating the things that you can't do by any other method. So of course you can redo structures that were previously known by crystallography and so on. But this one is a really good one. This is the work of Alexei Amunts, who was a postdoc with Venki Ramakrishnan, trying to work on the structure of the mitochondrial ribosome. Now, mitochondria inside, let's say, beef heart mitochondria, you get a lot of mitochondria in energy-needing tissues like the heart. However, and the mitochondria have their own ribosome, but there's also many more cytoplasmic ribosomes. So what Alexei was trying to do was to purify the mitochondrial ribosome, crystallize it, and determine the structure, which is similar to what Venki had been doing previously with other non-mitochondrial ribosomes. But he could never get it pure. This is about 80%. This image of the ribosome preparation, Alexei, is about 80% mitochondrial, 20% cytoplasmic. But Alexei got together with another postdoc from Shor's Sherez group, Xiaochen Bai, who's now in Dallas leading his own group. Alexei is now in Stockholm leading his own group. And they worked together and analyzed images like this and got initially a model that shows the average of all the structures of all the different ribosomes. But using software that Shor's Sherez has developed, rely on, which does allow you to analyze three-dimensional classes from your population of particle images. You can then ask to separate all those images into the ones that agree with one another more. And by doing that, they were able to find all of the images that were mitochondrial ribosomes and separate them from the ones that were cytoplasmic ribosomes. And there were others that were sort of a bit junky. But you can also see that some of the mitochondrial ribosomes are intact. Others have lost their small subunit. And so by using other features in Rely On, you can focus on the large subunit, refine all the orientations in all the particles and calculate a 3D map like the ones on the top right. And you can do the alignments by focusing on subregions of the structure, like the large subunit, and then separately from the same data set on the small subunit, and then end up by finding out the structure of all the proteins in that intact, ATS, 70S ATS ribosome. And I'll show just a bit of the density to show how nicely it works. It's 3.1 angstrom. And here is a little bit of Watson-Crick double helix of RNA that's in the core of the ribosome. Here would be one Watson-Crick base pair taken out of the double helix. Here is a little alpha helix, the same as the Linus Pauling prediction back in 1950. Here are little pieces of beta structure with a loop and side chain sticking out from it. So the cryo-EM method allows you to work with tiny quantities that are not pure and that do not have one conformation. So it's a very powerful method. So I'll just show you a couple more examples. Here is that same picture I showed earlier, mitochondrial complex 1. It's a membrane protein. You have to get it out in detergent. This was a preparation that Judy Hurst, who was a group leader and is now the director of the MRC mitochondrial biology unit in Cambridge. She came over about 5 or 6 years ago and said she had heard that cryo-EM was getting to be quite powerful. And she had been working on this for many years. They got crystals, but they didn't diffract well. They didn't have all the proteins in them. And so she and her postdoc, Shaipan Ju, brought it. Vinoth Kumar took images like this, and within a month or so had structure like this, 5 angstrom structure. At high contour level, there are 8 iron sulfur clusters that are responsible for the electron flow from NADH inside the mitochondria, flowing down to quinone to reduce the quinone in the membrane. And then in the membrane, there's 76 transmembrane helices pumping 4 protons across for every electron that flows down the redox chain. And so they carried on working with this. First at a lower contour level, that's the lipid detergent belt around it. But with about a year's more work, they were able to get a 4 angstrom map. And actually now these structures that are about 2.5 angstroms are very good. But they were able to build the structures of all 45 different polypeptide chains in the mitochondrial complex 1. The blue is the core, 16 core subunits that are homologous to similar subunits in bacterial versions of complex 1 that are about half the size, and that another group led by Leo Sasanoff in the same mitochondrial biology unit, had determined a bit earlier. So that all helped. And then here is that same structure in the top left. Now parts of it known, parts of it shown, so that you can see all the different. So for example, here is a central part, one of the protein translocation sites in the membrane with this ND2 subunit. So that's a structure that can only be done and has only been done for the large mammalian complexes by electron cryo microscopy. So a third structure I thought I'd show you is a step back to haemoglobin. And so Miriam Koschei was a student in Munich, in Martinsried, with Radostin Danev, her supervisor, and Wolfgang Baumeister. And Miriam said, I think we should take cryo-EM pictures of hemoglobin, 64 kilodaltons, 4 subunits. And both her supervisors said, no, it's a very bad idea, don't do it. So, but she ignored them and took images like this on the left. They had a slight enhancement over the normal methodology with a faceplate, but processed them. And early next week, she had this map, and you can see the heme group of human hemoglobin, the proximal and the distal histidines, just like Max Perutz and John Kendrew found in hemoglobin and myoglobin using X-ray crystallography about 50 years earlier. This was some of their alpha helices, showing how nicely the structures. So that's the third structure, and I've got 3 more to show. One is an enzyme that Vinod Kumar worked on with Ramaswamy in Bangalore in India, but took some of the images in Cambridge. It's called phenylacetic acid degrading enzyme, and they did it with and without the different ligands that are responsible for the enzyme reaction in degrading the phenylacetic acid. And the difference maps are difference densities shown in red. The gray is the density, and magnified at the bottom, you can see the octanyl coenzyme A density from the cryo-EM difference map and NADPH difference density from the cryo. So what this shows you is that it's possible not just to determine the structures of the proteins or the protein complexes, but you can also look at ligands and then go forward into drug design and help to develop better drugs in human health. And I'm just going to finish it by showing 2 structures now of complexes that are really being used to improve the drugs in different things. So this is a G protein coupled receptor responsible for signaling in cells. This is the adenosine A2A receptor. And this is images taken by Garcia Nafria in Chris Tate's group at LMB. And he's now back in Spain doing his own work again. And these are images taken with this same faceplate that Miriam Koschei took. These are 2D classes. Here is the overall structure of the G protein coupled receptor with 7 transmembrane helices, does signaling like 300 or 400 other molecules that are involved in this same family, signaling for different things in human physiology. And this is the G protein complex, trimeric G protein complex, with a nanobody helping to stabilize it. And then in the middle of this protein, in that red square, you can see this density. This is NECA, which is an adenosine analog that's an agonist for this receptor. And this is the density that they were used to get that model and to look at density. So that drug now, in principle, you know how it binds. You can then start to make modified chemicals and try to make a better drug. And a second one is this one, the serotonin 5-hydroxytryptamine 1B receptor, done in a very similar way by the same person in Chris Tate's group again. The images, the 2D classes, the resolution, 3.5 angstroms, some of the density for the helices, the overall density, and again, a magnified view showing the ligand, in this case, which is donitriptan, which is again an agonist that has some potential use in treatment of disease. So that's then given you a bit of a background into what cryo-EM is and why it's so powerful. But we can now take a step back and ask, what about the overall impact of cryo-EM in the whole world of structural biology? So the next slide shows a graph, which is a record of the growth of structural biology since about actually goes back to about 1960, when there was one structure, myoglobin, and 1964 when there were two structures, and then 1967 there were maybe 5 or 6. But now there are 170,000 different structures of proteins or nucleic acids or nucleic acid complexes or big macromolecules assemblies like ribosomes and complex 1 and so on. And the main growth has been in X-ray crystallography. That's the blue line here. So it grew from 1 every few years. And last year, there were perhaps, or this year, about 10,000 structures, atomic structures, are deposited in the protein data bank. And in the table, you can see the growth. So in 1997, there's about 1,000. And now it's sort of almost plateaued at about 10,000 a year. And then nuclear magnetic resonance spectroscopy, which I showed you one slide of, it grew and grew and was very popular in the turn of the century. About a quarter of the structures deposited were being done by an NMR, but it peaked in 2007, and since then it's declined. And now NMR is still very valuable. It's still used for drug design, but not for de novo structure determinations anymore. And then here comes the cryo-EM methodology, which didn't really take off till 2012, 2013. And you can see it overtook NMR. It's now going up with exponential growth quite rapidly. But still, we've only got 2,200 structures this year so far. So it's about 4 or 5 times behind in terms of numbers. But in order to know where to invest and what you should, if you're a young person starting in science, you have to decide which field, what type of work should you do. So one plot we like is the next one, where we do the ratio or the log ratio of X-ray over EM structures. And you can see around the year 2000, there were 2,000 times more structures that year being determined by X-ray crystallography than by cryo-EM. The microscopes and the equipment, the hardware got better, and it dropped to about 200 to 1 and was pretty flat until these new detectors that I mentioned came in about 2012, 2013. And since then, you can see the growth on a log scale is linear. So the doubling time is roughly for the cryo-EM is about 1 year and 8 months. And the extrapolation that we like to do is to put a straight line through that and then ask, at what point will there be the same number of structures deposited by X-ray and EM? And that's mid-2024. But that, of course, obscures the fact that most of the structures done by cryo-EM are a lot bigger than the structures done by X-ray. X-ray still has higher resolution, but it's probably 7 or 8 times more atoms and proteins. So you could already say that cryo-EM is the dominant method in terms of either the numbers of residues or the difficulty of the structures. And so it's definitely here to stay. And one of the goals now is to make it more accessible and more affordable, so that people who are in out-of-the-way places can do it just as easily as people who are in well-equipped labs. So that's essentially the end of the cryo-EM bit. But I promised Parvez I would end by showing a few slides about some of the structures that you find and how they impact on vaccine development and immunity and so on in the SARS-CoV-2 virus that was only identified earlier January 2020. And I've taken, on the left-hand panel, these are 2 images that you get in Wikipedia, who of course compete to make the best images. And on the top left is a schematic diagram of a SARS-CoV-2 virion, and it shows the 4 structural proteins. The virus genome is about 30,000 RNA bases. It codes for about 15 or 20 different proteins, but only 4 of them are structural. Other ones are enzymatic and so on. There's a spike glycoprotein called S. There's the envelope protein called E. There's another membrane protein called M. And then there's the structural protein that binds to the RNA, the nuclear protein called N. And the virus is a single-stranded RNA. And the RNA inside the virus provides the code, the message for making all of the components of the virus. And so, for example, it makes a polyprotein with lots of enzymes that a protease in the virus cuts up. And I'll say a little bit more about that later. But in the virus, and the bottom is an artist's rendering of it using some of the other information. And the structures you see are the same ones you see. So this is the red at the bottom is the spike protein, and these other membrane proteins are the ones that you see in different colors. So these are the ones that are often used in news bulletins and so on. But if you ask, how can you develop immunity against the virus? Obviously, you can be infected, make your own antibodies and recover. Unfortunately, about 1 in 200 people who are infected seem to, are seriously ill and die. So it's very important to develop better ways of treating it. And vaccination is the key one. And the only protein that gives you antibodies that can prevent infection is this spike glycoprotein S. And it has a complicated life of its own. It's synthesized as a single protein. When it binds to the receptor in your breathing tubes or in your lungs and so on, it binds to a molecule called ACE2, the receptor for the virus on cells, but also on the surface of the cells there's a protease, and it cleaves the spike protein. So when the vaccine development companies are making a vaccine, they have to be very careful to deliver the spike protein in the vaccine that gets injected into people in the right state. And so there's a lot of knowledge from earlier coronavirus work, particularly, for example, from McLennan's group in Austin, Texas, that the people working on this new virus have been able to take advantage of that. Anyway, that's sort of a bit of a background. So the goal of vaccination is to get a well-behaved preparation of the spike glycoprotein that you can get antibodies against, and that will then neutralize and block infection by other new viruses that come along. And on the right is one of the many studies that electron cryomicroscopy people have made using the cryotomology, the electron cryotomography methods, which don't need repeating structures and so on. And this is from one of the groups. This is John Briggs, who's here at the MRC, Laboratory of Microbiology, and one of the people in his group, Kay. And these are sections of 4 different virus particles. And you can see the membrane, you can see the RNA with the nuclear protein inside it, and then you can see sticking out from it the various spikes. And by doing the tomography on this, you end up with a model. And this is a real model of a real virus in a real preparation, isolated from the supernatant of cells grown in tissue culture. It's been fixed with formaldehyde, so it's not infective. And you can see that the spike protein can turn, it can tilt to the right, to the left. There are different structures of the receptor binding domain can open up. And these purple regions are the receptor binding domains, which is part of the spike glycoprotein, which is about 150 kilodalton protein, 1,200 residues, so it has different domains. And the spike adopts different structures. So it can be closed so that it cannot bind to the receptor, and then it can be open with one site available, 2 sites, and so on. And then once it binds to the receptor, it has a structural change following the proteolysis and fuses with the membrane of the host cell and releases the RNA into the cell, and then you're infected. And so this is a good overview of the structures of the coronavirus and what cryo-EM, in this case tomography, has been done to understand how it works. But you can also isolate the spike protein from the virus, or you can make it in vitro by genetic engineering. And here is some work, the one on the left by McLellan's group at Austin, Texas. This is the structure of the trimeric spike protein itself without anything bound to it, with the polypeptide interpretation on it. And the purple color, or the magenta color at the top, is the receptor binding domain of the coronavirus. And on the right is from recent work from a group in China, but working on, not on SARS-CoV-2. It's actually, I've typed this wrongly. This is SARS-CoV-1 with one of the receptor binding domains in the up conformation that you saw in the tomography, and bound to it is this angiotensin converting enzyme, the receptor from the host cell that the virus is about to infect. So there are now cryo-EM structures of the spike alone and the spike bound to its receptor. And then the next slide shows the structure of this same, on the left-hand side is the free SARS-CoV-2, and on the right-hand side from work from Pamela Bjorkman's group, where they've isolated antibodies from 10 patients who had been infected and survived and had recovered and had big antibody titers against the SARS-CoV-2 spike protein. And they've isolated them and done structures. And so here are two different states in which the spike protein has different conformations, and bound to them are the antibody domains isolated and, of course, cloned and expressed from the patients who'd recovered from corona. And so the goal of the vaccine, based on the spike protein, is to get you to develop in your own immune system versions of this antibody that you see here, determined by cryo-EM. And so that's the main, I think, hope in treating coronavirus. And these structures help you, but in actual fact, you cannot say that the structural biology has helped current treatments, because really vaccines, we had polio virus back in the 50s and so on. Structures are not needed to get good vaccines. You can do it. And all of the Moderna, the Pfizer, the AstraZeneca and so on, the groups that are doing this, they haven't really used the structure in the design of those the vaccine injections. They're designed by knowing a lot about the biology of it. But the one other hope involved in the coronavirus, there are the possibility of having small molecule drugs, inhibitors, pills that you take that can block the virus. And that's what's helped with HIV infection. Now there's a whole range of different small molecule inhibitors of the RNA polymerase, of the protease that cleaves up the polyprotein. And I'll just show you one of these. Now, there are lots of groups working on this, but this is a cryo-EM structure of the SARS-CoV-2 RNA polymerase that's responsible for reproducing the genetic material of the virus. Here's the structure done very recently by a group here at LMB, but there are many groups around the world who've done different structures of this. There are published ones with remdesivir, one of the inhibitors. But here's a cryo-EM image of the polymerase with a drug, favipiravir, bound to it using a new type of specimen preparation. But here is the density for the drug. And again, you see the environment. You can perhaps make it a little bit better. So I'm winding up now after about 50 minutes. I hope I've told you a little bit about an overview of structural biology, a lot more about the power of cryo-EM and how it's still in its infancy, and then a little bit about how the cryo-EM has been helpful in understanding a lot about how the coronavirus works, and as a result, lots of people are interested. So I'm going to finish just by showing some acknowledgements. Obviously, most of what I've shown you, some of it I've been involved with, and most of it is work by other people. So adenovirus from Jacques Dubochet, Phoebe Stewart, and Hong Cho's groups. Hepatitis B from Bettina Boccia, working with Tony Crowther and Hong Chu more recently. The mitochondrial ribosome, I've acknowledged all these people again. Alexei and Xiao Chen now have their own groups. Vinoth has his own group. India, Sweden, America, Complex 1. Hemoglobin, Miriam Koshayan now works for Novartis in Switzerland. The phenylacetic acid complex, and then these two GPCR complexes from Chris Tate's group here in Cambridge. And then in the SARS-CoV-2, I have to say a few days ago, I had to read a lot of the literature to collect these slides together. But Wikipedia is a very good place to start. And that artist's rendering was made by the Center for Disease Control in the USA by two people who are acknowledged on the Wikipedia site. The cryo-EM tomography work is from John Briggs's group here. The spike protein that I showed earlier is McClellan's group. He's worked on coronavirus proteins for 20 years in Texas. So he was well placed to add his group's power to the new virus as soon as it became available. And of course, the angiotensin converting enzyme, the receptor for the virus, was the same receptor for the earlier coronavirus. Pamela Bjorkman's done this work quite recently on the antibodies, and then there are many groups working on RNA polymerase drug design and protease, viral protease cleaving the polypropylene drug design. And if you look in the protein data bank, there are 1,000 or 2,000 structures done, most of them in the last year, all to do with coronavirus. So, you know, the method is being very helpful in that area. Okay, so with that, I'll stop and see if I can unshare my screen. Stop the screen and go back to being live. Am I still online?
Yes, I can see you, Richard. Thank you very much for presenting a wonderful lecture, really, really exciting, and giving a historical context to your research, acknowledging the work of many of your colleagues and researchers from around the world. Thank you for this presentation. And we have time for questions. I think we have quite a few questions that have come in. And also there was, I think, a technical glitch which prevented me from showing those slides, which actually I should show, because it really summarizes your achievement in very briefly. And I think I really need to show this, because what I'm hoping everyone is able to see this now. I'll just go back and make sure that it actually appears, and not I miss out again. So this is the slide that I wanted to show everyone in the introduction, because I think this is quite relevant. You can see that the beginning of his scientific life as a PhD student, Richard actually crystallized water-soluble proteins, which are relatively easier to solve. And I equate his solving of the structure of chymotrypsin, an enzyme, with a serine protease enzyme, with climbing and reaching the summit of Ben Nevis, which is the highest mountain in the British Isles. And as a matter of fact, as I mentioned, Richard actually climbed this mountain. So he has done both the crystal structure of this protein and climbed a mountain. Second, at the beneath, you can see I have a slide. Part of the slide here shows you the membrane proteins, amongst which is bacteriodopsin here, which is the protein that Richard crystallized and is the first membrane protein to have its express structure determined in a membrane environment. And that, of course, as I mentioned to you, is really, really the most challenging work that no man has ever been able to do, and Richard was the first to do this. So in the context of climbing mountains, it is like trying to reach the summit of K2, which is known as the Savage Mountain, which is the deadliest mountain to climb. And so really what Richard Henderson has done, he has climbed the easy mountains, if you want to call that, to the most challenging mountains. And I'm sure maybe he will try one day to really, really physically also climb Mount K2. But let's see if he manages to do that. Before I end, I need to really point out that there is a difference between climbing mountains and achieving the extra structures of proteins. With extra structures of proteins and electron microscopic structure determination of proteins, what you're doing is you are actually solving problems related to human health, designing new drugs and vaccines and so on. So from this point of view, it's an amazing achievement that Richard Henderson has done, and we are really, really proud of his work. And actually, to end the presentations today, we will have some questions that has been asked by the audience. And amongst the questions that has been asked is one by Professor Brian Austin, who is from St. George's Hospital Medical School in London. And he asks, is there a 3D structure of the COVID-19 spike glycoprotein yet? And I think Richard has already, of course, discussed this and shown that in quite detail. Do you have anything else to add, Richard, on this?
No, I think, you know, I spent quite a while reading the thing is, you could have a 1 week or 1 month meeting about coronavirus structures, and because, you know, because of the lockdown from coronavirus worldwide, many labs sent everybody home except for people working on coronavirus. And so consequently, many people said, I think I might work on coronavirus. So it went from nothing, or from very few people working on coronaviruses, because they weren't thought of as being terribly lethal. You've now gone to thousands and thousands of people beavering away. So what I showed in those 4 slides about coronavirus, they were just the tip of the iceberg, if you like, in terms of the volume of work that's been done. And there may well be lots of other better structures, but that was just to give you a flavor of it. In fact, there's many groups in China, because they were first on the scene. There are many structures for the protease, which actually has 3D homology to the chymotrypsin that I used to work on, and the RNA polymerase, as well as the spike protein in multiple conformations. And so we know a lot more about the coronavirus than we did at the beginning of the year. And so hopefully these vaccines will work, but the further structural information might make them better and better until instead of it being 70% or 90% effective, you might get them to be really 100% effective. So it's all very positive in terms of coronavirus at the moment.
Thank you, Richard. There is another question here from Professor Ahmad Amouche from Qadi Ayad University in Marrakesh in Morocco. And his question is, could we imagine to combine electron microscopy and microhandling, or even nano, of molecules, namely biological, and follow their interaction in real time?
Yeah, the thing about the power of the electron cryo-microscopy, I mean, you can think of new methods that haven't been invented yet, but cryo-microscopy, you have to freeze it. And if you don't freeze it, the molecules are moving around, they are more damaged by the electron beam, and you really can't do real-time with any resolution to talk of. So the way that you can do an analysis of, let's say, a progression of structural changes in a reaction is by freeze trapping. So in principle, you can do millisecond timescale, and you can time the plunging of the grid into your liquid ethane. You can often synchronize it with a laser flash or something like that, so that you make all the molecules into one state, and then you plunge it 0.5 millisecond, 1 millisecond, 5 milliseconds later. And then you can do trapping of kinetic intermediates, and then you can then infer in the way that crystallographers have been doing also for many years. You can make a story up about how it works, but you can't see how the atoms are moving in real time. You can see it at light resolution with optical microscopy, which is not so damaging. For example, by fluorescence, you can see things, but not so easily. But the electron microscopy methods are also being used for non-biological structures. You can do really good analysis of all sorts of microstructure fabricated arrangements. For example, future computers might well have nanometer length wires, and you can use electron microscopy in material science in that way. But the cryo is limited to when you plunge freeze it.
Thank you for this answer. I have a couple of more questions. One from Dr. Umakant, who is a colleague of mine actually from De Montfort University here in Leicester. And his question is, what is your esteemed opinion on ion identification using cryo-EM and X-ray crystallography? And how do they compare for structure-based drug design?
Yeah, so I think I'm going to assume that question concerns when you see the density in your map, can you tell whether, for example, it is a positively charged amino group at the tip of a lysine residue, or is it a neutral one? Or if it's an aspartic acid or a glutamic acid, which has a carboxylate group at the end of its side chain, is it negatively charged or neutral, or is it protonated and neutral? And in principle, it is true that the charge is sensed by the electrons as they pass through. And so a positive charge should give you a bigger signal than a neutral molecule. even though it differs only by one electron or one proton. And a negatively charged residue should give you, at lowest resolution, should give you a lower density. And people who do electron microscopy and compare it to X-ray crystallography maps have often wanted to interpret it in just that way that Uma Kanth has asked. However, there is a bit of radiation damage, whether it's from X-ray diffraction or from electron microscopy. You're irradiating it as you collect the data. And so there's always a little bit of radiation damage. And so the thing about radiation damage is it can often preferentially break the bond. For example, in aspartic and glutamic, breaks the bond between the beta and the gamma carbon. So the carboxylate falls off and you get less density. And in the past, people have been misled by radiation damage into saying that it's a negatively charged residue. So it's a little bit complicated, but there are new methods where you're using the power of the detectors and the computers to extrapolate back from radiation damaged structures, both in X-ray crystallography and in electron microscopy. You extrapolate the structure back to the point where before the radiation damage happened. But at this moment, there isn't really still anything all that convincing about it, but it's a hope for the future. So in both methodologies, there is hope for ion identification, but the question might be about, can you tell the difference, for example, between a sodium ion and a water molecule? And the thing about that is water molecule is H2O. So there are 8 electrons in the oxygen and 1 in each of the hydrogen. That's 10 electrons. Whereas a sodium atom, I think it's 11, you make Na+, that's 10 electrons as well. So often you can't tell the difference between a sodium ion and a water molecule in these maps, whether they're X-ray or... so you then have to look at what is that density near and what other atoms is it interacting with. And so there's a big database of structures and bond lengths. So usually the way you identify a sodium ion is because it makes a closer interaction to a nearby carboxylate group than a water molecule will make. In other words, the distance between the peak for what you might think is a sodium ion and the peak for the oxygen, and it's nearer when it's a sodium ion than it is for a water molecule. So it's a sort of indirect thing. And again, there are error bars on the accuracy of the peaks and so on. So it's possible, but it's complicated.
Thank you for this answer. I think there is another question here from Dr. Eid Brima, and he's honorary research fellow at De Montfort University. And his question is there any clear reason why X-ray is still a favorable technique?
Yes.
I think you're trying to change this. I've been
asked this a lot, you see, because
yes,
particularly the people who are on funding committees, they want to know, should they still put money into synchrotrons and X-ray crystallography? And the answer is definitely yes. And the synchrotrons are going to be upgraded. They're going to be 10 or 50 times brighter. They can do very little crystals. So even at the moment, when difficult structures and the productivity of cryo-EM, it's very quick, it's easy, everyone likes it. I'll give you one example. So recently we were over visiting the Diamond, the UK synchrotron for producing X-ray crystallography, and we had one of our microscopes there in a room kindly given to us by Diamond, by Dave Stewart and so on. And we were using that for developing better detectors in collaboration with a group at Rutherford Appleton Lab, Nicola Greeny. And one day, we were given half the lab. One day, we noticed in the other side of the lab, there are 6 people beavering away. There's a young professor and 4 or 5 students and postdocs. And so we said, where are you from? They said, oh, we are from San Francisco. And so we said, well, how come you've come all the way from San Francisco to this room at the UK Diamond Synchrotron to do the work. Can't you do this in America? They said, oh, no, this is a unique facility. And this is Frank von Delft runs this. What it is XChem, it's called. They have a computerized robotic system for mounting crystals and soaking them in 1,500 compounds to do screening for drugs by drug fragment screening. And so within a day, they make 1,500 crystals. Each one has a different compound in it. And then the next day, they're given time on the synchrotron, and they can collect complete 3D data, 300 data sets a day. So they would get, in 3 or 4 days, they get 1,500 structures. Now, by cryo-EM at the moment, even if you work hard, it takes you 1 day to get one structure. So at the moment, once you have to get a crystal first, but once you've got a crystal and you can soak compounds in, the X-ray crystallography for drug design is 300 or 400 times faster, and you get higher resolution quicker. And so there's no way. And also you'll be able to do smaller crystals. And there are new ideas also for doing certain types of X-ray microscopy. So we need the synchrotrons, but maybe we just need to keep upgrading the ones we have rather than building any new ones. And fortunately, synchrotrons like Diamond were designed with a very big diameter ring. So they've been future-proofed. So you just need to upgrade the magnets and the klystrons and so on to accelerate them. So there's a great future still. And so if you look at the average resolution done by X-ray crystallography, it's still higher on average than most of the cryo-EM structures. So cryo-EM can be improved, so can X-ray crystallography, so can synchrotrons.
Thank you. I have one final question that I would like to ask you, and that is about what do you think is the key message that you would like to give to the public, to the young generation, with respect to the huge amount of experience you had in your lifetime working in science? Is there any message that you want to give out that we all can benefit from?
Well, I think I remember when I was a student, an undergraduate, there was a book published in 1964, 2 books actually, Penguin series. It was called The World in 1984. And then it was, trying to look ahead to sort of George Orwell-ish future. And they asked something like 30 scientists and other futurologists to look 20 years ahead in 1964. And they completely missed, let's say, DNA sequencing, genome sequencing. They completely missed the power of computers as they grew. And so really, looking forward, you can really only look at the likely developments that are incremental compared to where we are now. But I would say there's a lot of enthusiasm for the development of what you might call, computerized pattern recognition, artificial intelligence, if you like. And you see this with AlphaFold, looking for patterns in protein folding. You can see it playing chess, playing Go. And so, in 30 years' time, there'll be a lot of advances in terms of automated computer-based technology that will help us to be more, have more labor-saving advances in knowledge and so on. But sometimes that question is asked to do with what do you, as an individual, think you should do with your life if you're a young person? And I have to say, my only view on that is that the most important thing is to do something that you find interesting and exciting yourself. And actually, Francis Crick used to say this. He said, why he was a civil servant or a operationals research person. He worked on war work during in the 1940s. But he said the thing he would like to do with his life and his career, he said the most important thing was the thing that you talk about in the pub or at coffee, the thing that you find most interesting and exciting yourself. And the reason for that is you are going to be more interested in it. So when things are difficult, you won't give up because you're intrinsically interested in it. And if you're interested in it, probably other people are interested in it as well. So I think there are two things. One is you can't predict the future unless you look only slightly further ahead, and then artificial intelligence is definitely the key at the moment. Obviously, human genomics, that's going to be very powerful. So at the moment, you can have your genome sequenced, and it doesn't help you very much unless you've got a monogenic problem. That's there are people with mutations in genes, and then you have to be careful. But in general, the multigenic parameter is not really very well understood. But doing something that you find interesting, and then if you think you've got as far as you can, you should move on to something else and then leave other people. So I can remember, again, in 1959 or so, we'd worked on this first serine protease. We said, oh, that's the end for serine proteases. No one's going to work on them. But there are now tens or thousands, hundreds of thousands of serine proteases, including the 3C protease in the coronavirus. That is, although it doesn't have a serine, it has a cysteine at the active site. It's the same fold. So this you couldn't possibly have predicted before. So, even areas that you think are now, you're going to leave it behind and move on, other people will find that interesting, and then new discoveries will be made that make that exciting to come. So, I think just do whatever you think you want to. I think that's the good one.
Thanks very much indeed for this wonderful answer to this question. And once again, thank you for taking out your time and coming to the Remote Fort University and making this presentation with an audience from around the world. And finally, what I like to say is that at Remote Fort University, we try to engage our students, our undergraduate students, PhD students in these lectures. And one of my colleagues, Dr. Joe Rushworth, actually engaged our students to enter a competition. And in this competition, they did some work describing your work, and we have a winner, and the winner of that competition is Parag Bhatt, and he's a second-year biomedical science student, and the runner-up was Josie Dunn, who is a first-year nutrition student. So these are students who have actually looked deeply into your research and were fascinated by what you've done, and they've actually produced some work which we judged, and my colleague, Dr. Joe Rushworth, has actually organized that, and I would like to thank her for this. So once again, Richard, thank you for coming and making this presentation, and we wish you all the best for your future, because I know you're continuing to work and continuing to do experiments. You are right now in the lab. We can see behind you all your folders and everything. So you're clearly very busy doing things, and the pandemic is not stopping you from your work. So this is a highly motivating for all of us, because many of us are going through difficult times. Some people cannot even go to the labs, others cannot go to work, and so on. So your enthusiasm and interest actually motivates us and gives us energy to do things, and we really appreciate your time. And we of course want to thank all the audience who have taken part in this event from virtually all the continents of this world, and this goes to show how much interest your work has created, and we hope that these people will again join in future events that Remote Fort University will be organizing. So thank you very much indeed, and I wish you all the best, and I wish you those who are coming up to Christmas, wish you a happy Christmas, and also wish you all a happy New Year. Thank you very much indeed.
Thank you, Parvez, and then we'll catch up in 2021.
Thank you very much indeed. Thanks. See you later.